Today we're going to be talking about new gene editing technologies for the treatment of liver disease. I'm joined by Dr. Clifford Steer from the University of Minnesota. Dr. Steer, welcome to the show.
Thank you very much.
So tell us a little bit about some of the major recent breakthroughs in gene editing technology.
Gene editing technology has been around for a number of years. It was actually sort of brought to the forefront back in the late 60s, 70s, 80s. It ultimately won Mario Capecchi the Nobel Prize for a gene editing technology called homologous recombination. But really what has come to the forefront of gene editing is CRISPR. And it's been around for probably about 3, 4, 5 years now. And it really has really made some incredible advances in terms of the ability to rewrite the genome, both for basic science and also for therapeutics too, in humans, animals, and plants.
So how do you see these technologies impacting our ability to treat liver disease going forward?
So the majority of liver diseases are monogenic in nature, which means that a single gene is involved. These gene editing technologies, particularly CRISPR, are basically unique in the sense that they can not only rewrite a genetic sequence, but they can actually replace entire areas of the gene with corrected versions. So it really is a remarkable step forward in terms of our ability to edit genes.
So how close are we? How close are we being able to use these technologies to treat our patients?
Well, I'd say very, very close. The majority of studies, of course, over the course of the last several years have been primarily preclinical in animal models, but there are human studies that are now being initiated in the eye and other organs, and it's just a matter of time and a relatively short time before gene editing will be applied to certain liver diseases. The important thing in terms about any success in gene editing for the liver, of course, is to be able to deliver your relevant molecules to whatever cell in the liver that you're targeting, be it hepatocytes, Kupffer cells, sinusoidal endothelial cells, or whatever.
So what's the future? What's next in gene editing therapies for liver disease?
I think the application, in other words, the delivery, testing it in a number of different animal models that include alpha-1 antitrypsin deficiency, hemophilia, Crigler-Najjar syndrome, and the list goes on and on and on. The complicated issues like NASH and AASLD and cirrhosis, these are more apologenic type diseases in which a number of genes are involved. And I think those will eventually be targeted, but for the immediate future, I think scientists will target those genes that are amenable to being rewritten in terms of their sequence and actually corrected.
Well, it sounds like you're really leading the way in the development of personalized medicine in liver disease, and we really appreciate you joining us on the show today.
Well, thank you very much.